my great pleasure to introduce Kate, who's the Director of the Institute of Behavioural Neuroscience at UCL and Professor of Behavioural Neuroscience and Wellcome Trust Senior Investigator. Kate is looking at how sensory information is assembled in the brain into complex cognitive representations, focusing on the cognitive map, which is formed by the hippocampus and associated structures. This underlies our sense of direction and our sense of place. This map is also used to organise the memories of life experiences. Kate, thank you very much.
Can you hear me okay? The microphone's on. So I'm a sort of slight wild card at this meeting. I think I'm probably the only neuroscientist. And it wasn't until yesterday that I looked at the talk title that got chosen for me and had a bit of a panic because I don't really work with AI, so I've tinkered with my title very slightly. So I'm going to actually talk about what I do work on, which is natural intelligence, but I am going to kind of say some slightly AI things in there. So there'll be a little bit. But what I want to do is to talk about some of the work that we're doing trying to understand how the brain conceptualizes space at the level of the single neuron, so neural networks, essentially, but real ones and not artificial ones. And then I want to kind of describe some of the work that's going on in my lab to try and understand how this map is put together and how it's used, and try and relate that a little bit to how people conceptualize the spaces that you guys build for them and so on. So, how does the brain conceptualize space? So just a very, very quick bit of neuroanatomy for those of you who don't know any at all. So the brain is divided up into a whole bunch of different regions. And so it's very modular. It's not like the neural networks that have been talked about here where everything just gets thrown into one big network and stuff happens. The brain seems to have divided things up into different areas. So, for example, can you see the pointer? So at the back here is the visual area. Here is a spatial area, but it's not the one that I'm going to be talking about. It's more to do with local space. At the front here is the motor system and the planning system. And then down here are a whole bunch of things that involve more cognitive functions such as language on the left side of the brain, processing of objects and assembling all of that into things like memories. So the brain has parceled all of these things out. I should say that those are the areas that are in the cortex, the outer part of the brain, but there's a lot going on underneath too. So if you peel off the outside, there's a lot of structural stuff underneath. Some people talk of this as the reptilian brain or the ancient brain or something like that. And to some extent, that's true because the cortex massively increased during evolution, whereas pretty much everything has got these same subcortical structures, or at least all vertebrates do. So the subcortical structures seem to have some very, very primitive functions to do with regulating things and organizing emotions and all that type of stuff. So we're interested in space. So there are different kinds of spatial behaviour, and as I just mentioned, there is one particular class of spatial behaviours that are essentially the behaviours for space that's within reach. So if I want to pick up a coffee cup, for example, I will be using my parietal cortex to figure out where the object is in relation to me and figuring out where all my limbs are in relation to me and the cup and organising movement from one to the other. And if somebody has damage to the parietal cortex, then they lose the ability to conceptualise that space. So, for example, they will eat the food on the right-hand side of their plate, but they won't eat the food on the left-hand side of their plate because they no longer have a concept of the left side of space. It's all pretty amazing, but that's not the kind of space that we're working on in my lab. So that was parietal lobe. Another structure that's important for spatial behaviour is to do with the kind of spatial behaviour where you're not really thinking. So if you're following a really familiar route from your home to the train station or something like that, and you've done it hundreds of times, and you're kind of driving and chatting and not paying attention, and you get to the station and you think, God, how did I get here? You know, didn't even remember the journey. So that's all controlled by one of these subcortical structures, the striatum, and it seems to take over. So once you've formed your map of space, you've gone to a new place and you've learned all about it and you've gotten pretty familiar with it, the striatum kind of takes over and says, look, leave it to me now so that you don't have to think about this anymore. And it organises our very habitual behaviours, including not just navigation behaviour, but also movements and things like that. So the striatum actually is the structure that degenerates in Parkinson's disease as well. Basically, it's involved in action, and it's reinforced action. So a lot of what the striatum does is noticing whether that was a good action or not. So there's the striatum, quite deep in the brain. So I'm interested in more long-range navigation, and that's not super long-range like migratory distances, but navigation of the kind that gets you across the room and out the door to the next room or something like that. And long-range navigation involves a system called the hippocampus, and it's the one that's recruited when you first go into a new place and everything is unfamiliar, or when you're in a familiar place and something unexpected happens, like you were driving on autopilot and then you suddenly encountered roadworks. You suddenly have to stop and dynamically replan what you're doing. So this is all taking place in the hippocampus, and this is one of the here it is down here, quite deep in the brain. I should say instantly, if you're not familiar with these, that this is the front here and this is the back. So the brain looks a little bit like a boxing glove, and the thumb points forward, so that this is the thumb here, and the hippocampus is in the thumb, as it were, in humans. And it's turned out to be really, really an incredibly important and interesting structure, so a lot of people are studying it. So we're kind of interested in space and how the hippocampus forms a map of space, and interested in this at the level of the single neurons. And what the neurons seem to be doing is representing the space. So just a very quick word about what that means. So it's a word that we bandy around a lot, and people sometimes query whether it means anything, but a representation basically is a kind of what we call an isomorphism. It's a structure that has some type of mapping with the thing it's trying to represent. So in the case of space, you create some kind of structure, like a map or something, or this little scale model. This is actually a little model, perfectly done scale model of a town in Holland somewhere. So it's isomorphic in the sense that each place in the real world has a kind of corresponding place on the model. But exactly the domain in which you make the isomorphism varies depending on the situation, and the nature of the isomorphism varies. So obviously the brain is not making a tiny little scale model of space inside of itself. It's doing the same thing, but it's doing it in a slightly different way. It's using patterns of activation instead of physical mirroring structures. And what we think happens is that the pattern of activation somehow represents the space in as much as whenever you go to the space or think of the space, this pattern of activation recurs. There are lawful relations between the patterns of activation. So, for example, if you go from one place to the one next to it, the activation moves from the representation of that place to the representation of that place. So there's the same kind of relationships in your representation as there are in the real world. So that's the kind of thing that we're looking for in the brain. So we call it a map because maps are what we call for representations of space, and it's isomorphic, obviously, in the spatial domain. So how do we actually study this in the brain? So the human brain is very large and complex, and there are quite a lot of ethical restrictions on what you're allowed to do to humans. So that really restricts what we can do in a quite unfortunate way. So what we've done is we've turned to our favourite laboratory animal, the rat, and in fact, more recently, the mouse, for reasons I'll go into in a minute. So rats are a really good study because they have pretty much all of the same things in their brains that we have in ours, except they have a much smaller cortex. So this is the rat brain here. It's about the size of a walnut, so it's really quite small. And it's quite smooth. So whereas the human brain is very wrinkled because it's tried to pack a lot of cortex into quite a small space, the rat brain is quite smooth and there's hardly any cortex. So that means that the things underneath are quite easy to get to. So what we're trying to do is to listen to the language of the neurons and decode the patterns of activation. And the way to do that is to physically put some tiny little wires into the part of the brain that you want to record from. So this is a cross-section of the hippocampus. So this is the hippocampus here. It's a really beautiful structure. It's made of these two interlocking cell layers that are kind of C-shaped. So the whole hippocampus is sort of the shape of a cashew nut, and what they've done is cut through it. And what you're seeing is the cross-section. And you can see that there are these two interlocking cell layers. So This one here is called the dentate gyrus, and this is the one that seems to receive most of the information coming in from the cortex. And it does something with that information and then sends it through fibers to the cornu aminis, which is this region around here, which does something more. We're still trying to figure out exactly what, and then sends that information out again. So what you can see here are the little blobs, which are the bodies of the neurons, and then these coloured tendrils, which are the axons of each neuron trying to kind of connect to these other ones. Axons and dendrites all mixed together. So what they've done here is label the neurons with a genetic marker that makes the neuron make a protein that lights up under fluorescent light. So this really amazing technology has come along with mice, which is why we study mice now, where you can actually tinker with the genome. So you can engineer a mouse that makes green fluorescing neurons or something like that, or indeed an entire green fluorescent mouse, they sometimes do. So then when you put the brain under a microscope, you can see it. It's very beautiful, as I say. So it was discovered a few years ago that these neurons, using this method, which was developed partly by John O'Keefe, who was my old boss, my old postdoc advisor. So what he did was develop this method of putting the electrode into the hippocampus. And he did something that no one had ever done before, which is do this in an awake animal. So the animal was anaesthetized to have the electrode implanted. And it had a little connecting device cemented to its skull so that a few days or weeks later, after it had recovered from the surgery, you could then connect up that connector to a computer. And you can collect the action potentials. So where the wire is sitting next to the neurons, the neuron produces action potentials, which on the oscilloscope look like these little kind of blips here. So they're called spikes, for obvious reasons. They look like spikes. And what we do is we pick off each spike and then say, when did this spike happen? And when did it happen in relation to other spikes? And when did it happen in relation to what the animal was doing or thinking about or whatever? And he did this, listening to the spikes through a loudspeaker as the animal was walking around. And he found that in the hippocampus, if you listen to one of these neurons, all of the spikes from a given one of these neurons appears in one particular region of the environment. So, for example, they would all congregate here in the southeast corner of the box. So he proposed, along with his colleagues, that the hippocampus is trying to form a map of space, and each of its neurons has a little piece of the big picture. It's kind of like a little pixel map, if you like, for space. So this is, if you let the animal run around for a while, this is what you'll eventually get. You'll get a blob that represents the place in that environment where the cell was kind of interested and activated. And that's called a place field, that place where it becomes active. And hopefully, if this video works, you can actually hear one. So helps if I unmute my... So you're listening to the spikes, and the animal's just walking around a box. You can see the recording cable. And when it goes up, it's just sitting there, not much is happening. Now, hear that sort of machine gun sound? That's that cell getting kind of interested. This rat, unfortunately, spends a lot of time not in that place. But if you'll hear in a minute, that he'll go back. There we go. So every time it goes back there, that cell starts up again. And if you listen to him for a long, long time, then you would eventually hear that, that all of the spikes were occurring in the north part of the box. And if you were listening to a different cell, it would be a different part of the box. So actually, if you listen to several cells, you find that they more or less tile the space. And this is how we typically represent the activity as a kind of a heat map of the sort of activity of that cell. So you can see that these four neurons that were all recorded at the same time with a bundle of electrodes, each cell has a different place in that box. So although the cells are right next to each other, the places that they're interested in might be quite a distance away. So that kind of suggested that the hippocampus is trying to form a map of space, and then people got interested in trying to test this idea, and a whole lot of experiments were done. But the one that has become most famous is this experiment by Richard Morris, who was my PhD advisor. So he developed this task called the water maze, and what it is a big swimming pool. And the reason that it was such an interesting task is that because the animal is in water and swimming instead of walking around, it can't be using scent or anything about the texture of the surface or anything like that to know where it is. It's got to be using information about what the animal can see in the room around it. And he found that if you make... sorry, that's a picture of the rat swimming. So when you first put them in there, they swim for their lives, panicked, as you would. And then eventually they bump into a little platform. And then they can climb onto the platform and escape. And then if you put them back in, they'll be much quicker to find the platform. And then after 3 or 4 times, they'll swim straight there. So that tells you that they know where the platform is and also that they can plan a route to get there. And what Morris found was that if you make a hole in the brain where the hippocampus is, in other words, damage the hippocampus, and then take the platform out, whereas a normal animal will search where the platform was, one without a hippocampus will just search aimlessly. So it looks as though it doesn't know where the platform is. So that was really good evidence that the hippocampus has some kind of a map of space. And then people took this a little further and said, well, what about humans? Is that true for humans as well? And this was done over a number of years by a whole bunch of people, but some of the most important work was done by Eleanor Maguire at UCL. And she found that, so she developed virtual reality methods of getting people to navigate, and she found that people who had damage to the hippocampus couldn't find their way around these virtual environments, and that when normal people navigated, the hippocampus became active. And then in the experiment that she's most famous for, which really annoys her because it's not her best experiment, but it's the most eye-catching one, what she did was she just took a whole bunch of London taxi drivers who navigate all day long for hours and hours and put them in the scanner and just measured the size of their hippocampus, and that she found that they had enlarged hippocampuses relative to... So part of the hippocampus was enlarged. Actually, the other end of it, the non-spatial end of it, was actually slightly shrunken. Nobody ever mentions that. But anyway, the spatial end of it was enlarged. And furthermore, it was more enlarged the longer they'd been driving taxis. So it wasn't just that people with good hippocampuses become taxi drivers. It looks like if you practice using your hippocampus, then it will grow. So some of that work was done by Hugo Spears, who has gone on to... set up his own lab, and he's very interested in this whole navigation system and has been extending that quite a long way. And then finally, people did manage to record place cells in humans. So while it's not ethical to just take a person and make a hole in their head for no reason, there are a whole bunch of people who have electrodes implanted into the hippocampus to monitor for epilepsy. So these are people who are going to have surgery, and they want to identify exactly where the focus is so that they can destroy the focus without destroying any other bits of brain. And so while the electrodes are in there for recording the epileptic activity, neuroscientists have come along and said, look, could we just listen a little, kind of eavesdrop on what's going on? So they got people to do a virtual reality taxi driver task, so a little bit like what the London taxi drivers were doing, except a far simpler environment. So they're kind of navigating around this grid-like street, so it's the most boring city in the world. It's just this kind of 3x3 grid. And from the point of view of the taxi drivers, you can see the various buildings and things. And what they have to do is to pick up a passenger at one place and then take them somewhere else. So they have to plan a route. And what they found was that in the hippocampus and in regions right next door, single neurons in that region became active in certain places in this virtual environment. So, I mean, it's amazing that it worked, but it worked quite well. So you can see these hotspots. So this cell, for example, likes to fire over here. You can see the red kind of hotspot pixel. So it looks like it's not just a thing in rats, that humans also have this system, and it seems to be for space in us as well. So people got very interested in this discovery of place cells, and then the hunt was on for how does the place cell know where the rat is? What information is the place cell getting? How's that getting in? So people started recording from areas around the hippocampus. So here's the rat brain. The big blue thing is the hippocampus, which I said was like a cashew nut. So there are some other little areas of the brain in the cortex around about, and it was found by this guy here, Jim Ronk, that if you record from, his discovery was in this area here, the postsubiculum. If you record from this area, you find cells that only become active when the animal faces in a particular direction, but they'll fire anywhere in that environment. So they don't care where the animal is, but they care which way it's facing. So it's a little bit like a compass. And you can show that the cell is responding to the information that the animal has about direction, because if you have the animal in a closed environment with only one landmark, if you move that landmark, so for example, here's a white piece of card that's the only thing that the animal has to tell it which way around it is in that box. If you take the animal out, move that card and put it back, then the cell will rotate the direction that it likes to fire in. So it's not a geomagnetic detection system. They're not responding to magnetic north. They're responding to local visual information. And these head direction cells, they kind of got a bit neglected for a while because people were really interested in place cells, but interest is gradually increasing in these because it's a window into the processing that's going on that underlies all of orientation, and it turns out to be really, really kind of critical and fundamental. So some of the things that we found out about head direction cells are quite informative for how we conceptualize space. So one thing is that they use information about the environment, but they much prefer distant information. They don't really like information that's close by, and particularly things that you can walk around are not very useful. Whereas if something is a long way away, it's directionally really stable. It always has the same direction relative to you. If something is close by, like a chair or a door or something, then its relative direction, the direction in absolute space that it's trying to encode, trying to signal to you, changes because its position relative to you changes. So it's not so good, which makes a lot of sense. The other thing that the head direction system doesn't like is rotational symmetry, because if you're in an environment where it kind of looks the same, whether you look this way or look that way, and a really good example actually is this building in this circular little bit of street. I don't know if anyone else is getting horribly confused, but I keep getting the entrances to this building mixed up and forgetting whether the stairs go this way or this way. Because there's rotational symmetry. Like I came in one way the first day and I came in the other day the second day, and it looked kind of the same. So the head direction system really doesn't like rotational symmetry. Unfortunately, architects seem to really love rotational symmetry. and that plays havoc with our ability to find our way around. So it's just a plea out there for... So the head direction system doesn't mind mirror symmetry, actually. So because that's not ambiguous in terms of direction. So it's nice and symmetrical, but it's also, you can remain oriented. So if you're desperate for symmetry, go for mirror symmetry. Okay, so then the third big discovery in this area was made sort of about, it must have been 30 or 40 years after place cells, and it was an experiment that was done for the same reason as with the head direction cells. It was, how do the place cells know where the rat is? And these people, the Moses, recorded from this area here, which is called entorhinal cortex. And they recorded from here because the entorhinal cortex sends... So it sits there at the back of the brain, and it's collecting information from most of the rest of the cortex and sending it into the hippocampus. So it's like this kind of funnel for information coming into the hippocampus. And this particular part of it, which is in green, the dorsomedial entorhinal cortex, projects to the part of the hippocampus where all the place cells were. So the anatomists that they collaborate with said, why don't you put your electrodes there and see what you can find? And they did that, and they found something really remarkable, which is that there are cells which produce these hotspots of activity, just like place cells do. But instead of producing one hotspot of activity, they produce multiple hotspots. So this is one cell, and it's firing here in this big box, big 2-meter box. But it's also firing here, and it's also firing here. And if you look at those blobs, they're all the same size and shape, and they're all the same distance apart, and they're lined up in these nice rows, and they form what's called in mathematics a hexagonal close-packed array. So it's this remarkably orderly arrangement of hotspots of activity. And that immediately makes you think, what on earth is going on? So when this paper came out, this was one of those flashbulb memories for many of us in the field, because nobody had ever seen anything like this come out of the brain. I mean, mostly what comes out of the brain is random junk, stuff. Every now and then, after a decade or two of work, you find some signal that you can kind of get a handle on. But something as regular and beautiful as this has just never been seen before. So it really electrified the field, and it suggested to a lot of people that what these cells are trying to do is to form a kind of a grid reference that lets the place cells know about directions and distances, because these cells actually project to where the place cells are. So they named these cells grid cells, and they have just been a very, very kind of stimulating discovery for the field. And now we're trying to find out what they do. So O'Keefe and the Moses got the Nobel Prize in 2014 for the discovery of place and grid cells. And I really think they deserved it because they were amazing discoveries. So just to recap, we've got place cells, which seem to be the sort of you are here signal. We've got head direction cells, which are you are facing this way signal. And we've got grid cells, which seem to, they sort of tell you where you are, but only in a very coarse way, because if a grid cell is firing, if only one grid cell is firing, you could be in any one of a number of places. But actually, if you've got enough grid cells firing, then it turns out there's only one place that you could be. So actually, the grid cells do have quite a lot of spatial information. And then there's one other cell type, which I didn't mention, which was also found in the entorhinal cortex along with the grid cells, which is these border cells. And they fire when the animal gets to a wall. And it doesn't matter in which environment, any wall, but it does matter the direction of the wall. So it might be any north wall in any environment at all, or any west wall, but only north wall or only a west wall. So what are grid cells for? So we don't exactly know, but one possibility, and that was the immediate hypothesis, was that they might be for path integration. So path integration is this phenomenon that's very familiar to us all, which is that you could be walking around not really looking at landmarks, but you sort of know where you are still, and the reason is that you're tracking your movements. And this has been shown really elegantly in a whole bunch of animal experiments, and some of the early ones were done by Rudy Weiner, who studies ants. And he found that if you have an ant which leaves its nest, it'll go off foraging. I don't know how well you can see because this is an old photocopy, but you can see this kind of curly, tortuous path. So it's hunting around for food. And this is a particular kind of ant, a desert ant, and they can't stay out too long. So once they find food, they have to go straight back to the nest. So what happens when they find the dead fly or whatever is that they will then turn around and they'll go straight back to the nest. So that straight line is the ant making a beeline, as it were, straight for home. And Weiner found that, so you would think, how does the ant know how to get home? And initially people thought, well, it must be able to smell, it must be some olfactory signature. But Weiner found that if you pick the ant up at the point where it's just got the fly and move it 2 meters to one side, then the ant will go home in the same direction and the same distance, but 2 meters away. So it'll end up 2 meters away from the nest. So clearly the ant had in its tiny little brain some kind of vector for home. It wasn't smelling out the nest. It just knew the distance and the direction back to home and kind of fooled it. It wasn't evolved to be picked up by experimenters and to know what to do. So that process of constructing a vector as you kind of walking around by tracking your movements is called path integration. And that involves integrating both the direction that you've gone and the distance that you went in that direction. So you need to be able to put both of those things together. And grid cells seem like they might be the fabric of this because they are having to integrate both distance and direction in order to make their nice little grids. So they must know something about both the direction to be able to lie the blobs along this line, for example, and also they have to know how far 30 centimetres is or whatever. So they might be for path integration, and that would enable, for example, the ant to get back home, or indeed the humans to get back home. So we do this too. This was an experiment that was done with Hugo Spears and Claire Worsley and some other people, and Eleanor Maguire, I think. So what they did was they got people to walk blindfolded on a kind of an L-shaped outward journey, and then they had to walk back to the start blindfolded. So we humans are quite good at path integrating as well. So how do grids come about in the brain? We don't really know. There's a lot of debate. There's a lot of models out there. In fact, there seem to be maybe 10 modelling papers for every data paper in the grid cell field. But one thing that's come along in the recent kind of couple of years is that they might just form spontaneously as a result of networks getting information about location and about speed of movement. And for example, this paper, which came out a couple of years ago from Dori Derdikman's group, found that you can create a neural network that's taking as its input place cell information. So we know that the place cells, one of the places that they send their outputs to is back to the grid cells. So we've got a big sort of network here. So if you feed the place cell or simulated place cell data back to your simulated grid network and inject some movement information in, then you get things that look like grids. And what they're doing, what the network is doing, is essentially principal components analysis on the place cell signal. So it may be that that's what's happening, some kind of process like that. Similarly, there's a paper that just came out a couple of days ago from DeepMind, using a neural network that was using some kind of supervised learning algorithm. And again, it was taking as its input information from the movements of the animal and so on. And the simulated animal had to do a sort of a path integration task that had to find its way kind of back to the start. And they found, again, that the grids emerged naturally as a result of these interactions going on in the network. And they also had shown that you could, in theory, use something like this to compute the homing vector. So that was pretty cool. Whether that's actually what's going on, we don't know yet, but at least it's a kind of proof of concept. So we've learned quite a lot of things from studying place and grid cells. I haven't got time to go into all of it, but one thing we have learned is that the boundaries of the environment are very important, at least for rats and probably for lots of other animals as well. And one of the reasons, I think, is that boundaries provide very good spatial information. So this is one of the first demonstrations of this in grid cells, and this was the paper from my lab, which was the first lab that wasn't the Moser lab to publish a grid cell study. So Caswell Barry and Robin Heyman. Caswell was a PhD student of me and Neil Burgess, and Robin was a PhD student of just me. And when the grid cell paper came out, we all powered off to the pub and spent a couple of hours discussing the grid cell paper and going, God, this is amazing. Why didn't we discover these? You know, blah, 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 blah. What should we do? You know, and the thing that emerged from that conversation was to do an experiment that Neil Burgess had actually done with John O'Keefe quite some time before with place cells, which is to put the animal in an environment and then deform it so that it was slightly rescaled and see what the grid did. So what the place cells had done, what the place cells had done in that situation is that they had sort of deformed their fields so that if you stretch the environment, the place cells seemed to kind of stretch as well. And different place cells would stretch differently. So they tended to stretch to follow the wall that was closest to where that field was. So different place cells would stretch in different directions. So it was quite a surprising discovery. Now, our initial assumption when the grid cells came along was that the grid cells must be telling the place cells what to do. And that seemed a bit weird because if the place cells are stretchable, but the grids have this nice regular structure that's presumably derived from the movements of the animal, how can both of those two things be true at the same time? So we said, well, let's see what happens if you stretch the box. Will the grid cells resist that, or will they stretch as well? And Caswell and Robin did a lot of experiments, and they showed pretty convincingly that the grid stretches as well. So it seems as though the grid is somehow attached to the walls, in as much as when the animal's walking around, the grid cell is somehow getting information about how far the animal is from the walls. And so if you stretch the walls apart, the grid cell is trying to use those walls to kind of know where to position all of its action potentials. And it's also using the movements of the animal, and those two things are kind of conflicting. And the resolution of that conflict is a slightly stretched grid. So the stretching is about half the amount that you actually stretch the environment by. So anyway, what that tells us is that the grid cells are really interested in the boundaries. That also tallies with a whole lot of animal experiments showing that lots and lots of animals are interested in boundaries too. And this is actually a video that we took upstairs of ants. So I don't know how well you can see them. They're just here. What's the sound on here? There we go. Take away the conversations. You can see the ants, and what they're actually doing is they're trying to get back to their nest, which is up here, I think. And they've got this whole arena. You'll learn more about this later. This whole arena they can be walking around in, but they've decided to go down the wall. So this is the same for mice and rats and indeed humans. Like lots of animals don't like being out in the open. They like to be near boundaries, and the brain has decided that that's a really good anchor for the spatial representation as well. So just to kind of finish telling you quickly about some of the current projects that we have going on in the lab, and basically we're interested in these cell types and how do they work and how do they know what to do and what's the animal using them for. So one thing that we're doing is what you might call generative models. So we've heard a little bit about neural networks that create things. So it looks like the place cell network can do a bit of generating of its own. So as the animal is walking around, the place cell is collecting information, and if it gets the right information, it will fire and produce a place field, and that's fine. But when the animal is just sitting there not doing anything, it goes into this sort of different kind of state where you get bunches of cells all seeming to be active together. And if you look at which of the cells are active while the animal is just sitting there, you find that very often it's the place cells that should be active in this environment if the animal was walking through it. And this is a phenomenon called replay. And here is an example. This is an example, sort of schematic, of an animal that was walking this way along a kind of a plank, a linear track. And you can see that this cell here produced some action potentials, so it's kind of coded in orange. And then this cell started up and started producing green ones. So as the animal was walking along, first the orange cell fired and then the green cell fired. So those are the place fields. Then when it went back the other way, because the map isn't reversible, different cells fire going back the other way. So the two new cells fired, this one, this blue one, and this yellow one. Now, if the animal is just sitting there and you look at the cells that are active, you see patterns. For example, you'll often see the orange cell firing and then the green cell firing. Or, the blue cell firing and then the yellow cell firing. And you can see long strings of these that correspond to the long strings of place cells in the environment. And it looks very much as though the animal is sitting here thinking, where am I going to go? I'm going to go there. So if I go there, I'll encounter this place and then this place and then this place and this place. So the place cell system seems to be simulating the consequences of moving in that direction. And then there are some other weird things too, like sometimes you get sequences but they're going the other way as if the animal was moving backwards down the track. And we don't know why this is happening, but we think it's something to do with trying to reinforce connections that are relevant to the journeys that the animal might have to make. So what we're doing with this experiment is we've got the animal able to make several journeys from any one of these places to any one of these other places. And as the animal is sitting here, or at the start, thinking about what it's going to do next, but unable to move because there's a kind of an obstruction in the way, we're hoping that we'll see sequences of place cells that correspond to the journey that the animal is planning to make. Or indeed, maybe the other one. Who knows? We don't know what this would be for. So that's one experiment that's being led by my postdoc, Eleanor Duval. Second thing that we're really interested in is whether this map, the grid cell map and everything, is kind of extends throughout three-dimensional space, or is it really just flat? Like, does the brain just care about the surface you're walking on? Or, if we could float or fly through a big volume like this, for example, would we see that there's a map of large-scale space? So, for example, would we see that the grid cells form a three-dimensional hexagonal close-packed array? Or would we see other sort of possibilities and so on? So I have a postdoc, Roddy Greaves, who's doing this, and he's built this big kind of lattice, which is just a climbing structure that the rats can climb through. And instead of using a wire, as we normally do for these recordings, he's using wireless recording, which is now getting quite sophisticated and is small enough for a rat to carry. And these rats are kind of just climbing through. The third project is quite a big one, so it's involving quite a few people. And basically we're using humans and rats, and with the humans we're giving them virtual reality spaces. It's the most boring building in the world, and this is a kind of a top-down view of it, and it's got quite a lot of rotational symmetry of the kind that I said was really deadly for head direction cells. We've discovered that there's a population of head direction cells hiding away in one part of the brain that seems to be able to resolve these ambiguities. So, for example, this room here has, if you go through this door, then it has two doors on the right, whereas this room, if you go through this door, it has two doors on the right, and it looks exactly the same. And the only way that you know that it's not the first room is if you have an overall sense of direction. So we found that there are some cells in the brain that have the overall sense of direction, and there's a bunch of other ones that actually rotate to kind of with the room. And we think that some of those cells are kind of the big picture, and some of the cells are the small picture, the local, what you can see. And their job is to talk to each other and try and figure out what is going on. So it's a big project trying to unravel all of that. And then the final project is looking at what happens when an animal is trying to plan an escape. So, for example, if somebody shouts fire and you run for the exit, or in this case, if you're a mouse and you're exploring and then something frightening happens and you run for safety, where do you run to and which part of the brain computes that trajectory and other place cells involved? So this is a video taken from underneath a box. This mouse is just exploring. And over here is a sort of a safety, that's his little safe home. And at some point while he's exploring, this overhead screen will suddenly produce an image which expands rapidly, and for a mouse that's like a hawk coming at it. They really hate that, and they run like hell. And we're trying to understand, will it run for the door, or will it run for the place of safety, which is over here? And if there are barriers in the way, does it know to run around the barriers, or does it just run smack into them in a panic, and so on and so on? And there's part of the brain that we think we know might be involved in this called superior colliculus, and so that project is looking at that. Okay, and then in future we're going to expand and look at the bigger picture of not just the space that you're in, but how does the brain make a coordinated map of all of the linked spaces that you encounter. So that's a little kind of window into what we do, and I'd like to thank you for your attention and acknowledge those people.